Good morning all. Today we will be studying about gene therapy. So what do you know about gene? So we all know that gene is the physical and functional unit of heredity. So genes are made up of DNA. So we all know that there are so many genetic disorders which is not able to cure or even control, and it can pass to the other generation also. So by implementing the gene therapy, we can prevent or we can cure the disease, genetic disease, and also we can prevent transformation of this disease to the other generation also. So gene therapy is the introduction of genes into existing cells. Introduction of genes means the genes which we are prepared normally. Genetically means by genetic engineering we will be developing normal genes which is already abnormal in the patients. So gene therapy is the introduction of genes into existing cells to prevent or cure a wide range of diseases. It is a technique for correcting defective genes responsible for disease development. A person is having genetic disorder due to his gene abnormality. We will correct one gene and we will transform the corrected gene. Corrected gene is here termed as therapeutic gene. Therapeutic gene is inserted back to the person, and this therapeutic gene will produce the normal gene instead of the abnormal genes present in the body. So involved, the first gene therapy experiment is done in 1990 in US. A person named Ashanti De Silva undergone gene therapy for the disease ADA-SCID, adenosine deaminase severe combined immunodeficiency syndrome, and we can say that it was a successful, not completely, but it went well. So definition of gene therapy: it is an experimental technique that uses genes to treat or prevent disease. Otherwise, gene therapy includes approaches that correct the genetic defects or manipulate genes to induce tumor cell destruction in the hope of preventing or combating the disease. So here we can correct the genetic defects or we can manipulate the genes so that it can damage the tumor cells. There are two types of gene therapy: somatic cell gene therapy and germline gene therapy. So you all know that what is somatic cells. So what is somatic cells? These cells are any of the cells other than sperms and egg. So somatic cells are any of the cells in the body other than sperms and egg. So here the therapeutic genes is transferred into the somatic cells. For example, a person is having genetic disorder related to blood cell. So we will transform the therapeutic gene into the affected blood cell. Likewise, bone marrow cells, skin cells, etc. Here it is somatic. It will not be inherited into the later generation. A person is having a genetic disorder related to blood. Okay, we have done gene therapy and the condition is been cured. But the same genetic disorder can come for his children's because it is not inherited to the other generation. So that is the difference here, somatic cell gene therapy. So at present all the researchers are directed to correct these genetic defects in somatic cell. And the other type is germline gene therapy. Here the therapeutic genes is transferred into germ cells. So what are germ cells? Germ cells are either sperm or eggs. So therapeutic genes are introduced into eggs or sperms. And here it is heritable. When a person is having genetic disorder related to egg and With the help of germline gene therapy, we have corrected this particular genetic disorder means it is heritable. The same disease will not occur for that patient's children. Okay, it is heritable and passed on to later generations. So here, for safety and ethical technical reasons, it is not being attempted at present. Next is about the approaches. There are two approaches for gene therapy. One is in vivo gene therapy and ex vivo gene therapy. In vivo means direct delivery of genes into the cells of a particular tissue in the body. Ex vivo gene therapy means transfer of genes to cultured cells and reinsertion. While doing these both procedures or both approaches, we need vectors. Even in vivo and ex vivo, we need vectors to do this approach. So what are these vectors? So vectors are the one which is acting like a carrier. So to transfer the desired gene into a target cell, a carrier is required. Such vehicles of genes delivery are known as vectors. There are two main classes. One is viral vector and non-viral vectors. Retrovirus, adenovirus, and herpes simplex virus are examples of viral vectors. DNA lipid complexes and artificial chromosomes are examples of non-viral vectors. So remember, vectors are the carriers of the gene. The therapeutically made gene is entered into these vectors, and these vectors will carry this therapeutic gene and enter into the abnormal cell, and it will release the therapeutic gene into the cell.